Hello everyone! The Research Change program of IFMSA has the objective of giving students an opportunity of improving their skills in research in other learning settings. This year, Nova Medical School Local Committee, IFCM, is organizing the first SCORE Symposium, with the goal of giving our exchange students an opportunity to present the work they've done during their exchange, and, at the same time, encourage and promote medical research among national and international students community. So, without much further ado, we are honored to start the first SCORE Symposium with our first lecture, the role of research in clinical practice and how to conciliate the two, by Professor Frederico Bastos Gonçalves.
Good morning. Well, first of all, I'd really like to thank the organizers and especially Dr. Joana Azevedo for really setting the stage for this this exchange program. I think it's a really great opportunity to help medical students become more involved in research, for them to understand the difficulties and also the advantages of doing research. So I prepared a little, and especially sort of a talk for you, Joana Azevedo, that this is for really more setting the stage. I hope. So, okay, there it is. So this is me. I'm a faculty here at the University Hospital, and I'm also a consultant vascular surgeon. And I've been blessed with the opportunity to do a lot of research in my past. So I think I'm reasonably qualified to give you a sort of overview of this duality between research and clinical practice. I really like my clinical practice, but I also really like my research. So the first real question here is, why would you want to be a doctor in the first place? Can anyone give me one reason why they want to be a doctor? Anyone here? To help people. Okay, that's a good, but you could also be a humanitarian worker or ambulance driver. Why doctor? Because you want to help people, and probably because you're also intellectually stimulated to be to do things that involve your brain a lot. So that's because you're capable of doing that, and then you go into medical school with the hope that you will help people. So that's, for me, is also the reason why I do research, because not only you're helping someone in front of you, and that's your practice when you're one-to-one with the patient, but you're also helping a lot of people by doing something that improves the treatment of a disease, for example. So that's two ways of looking at being a doctor. And if you're a clinician, you can do both, and that's really good. So you can do the two. Everything we do right now in medicine is hopefully supported by some sort of evidence. These are probably, you've heard of this, the Oxford levels of evidence. This is the last revision of 2009. And at the very bottom of this layers of evidence, is expert opinion. So really, what you're being taught by your master is the lowest level of evidence possible. And what we do in research is we try to elevate this level of evidence with something that is structured and supported, that you can then use in your practice, not because your master taught you, but because it's proven to be true. And that's what this research is about. So really, research, as defined by the entities responsible, is a systematic investigation, including development, testing, and evaluation, designed to develop or contribute to knowledge. So this is research. When you're involved in research, you're hopefully expanding knowledge. You're creating more knowledge. Why would institutions like to be involved in research? Because it's been demonstrated that research institutions treat patients better. If you're doing research, you're doing your patients a good favor. You're doing good for the patients. And this is just one simple example of a study where they took a lot of patients treated with colorectal cancer, okay? And they found out that if you're treated for colorectal cancer in an institution that does research, you're more likely to survive the operation and you're more likely to have a better long-term outcome than if you're treated in an institution that does not offer research. Okay, and this is throughout all the fields of medicine in some way or another. It also improves quality of the service. Not only the outcomes are better, but patients feel they are more confident in treatment when the staff is involved in research, because everything is more carefully thought of. Patients are better informed, they know more about their disease, they can judge, they can do better informed consent. And this, in a way, will translate into the perception of health for the patient. As I said also, there's all sorts of evidence that will tell you that mortality is lower, quality of life is better. So research also does good things for the patient. And one aspect that I didn't mention here, but it's quite obvious, is that if you're doing research, you're probably more likely to offer patients an innovative or experimental treatment. This does not always work for the benefit of patients, but many times it does. So you're really giving the patients the chance to be at the higher level of medicine. That's what you get from doing research. So it's really a triangle here that feeds from each vertex of the triangle. So you have practice. Practice generates questions, generates needs. Then you have the research that feeds from practice and also from your thoughts, from theory, and integrates them back into the practice and also the theory. So it's really a triangle of activity that you need to be involved with if you're doing research. So this is something I really like. It's not my own, but I think it's a really great graphic representation of what this means. So let's imagine that this blank circle is total knowledge of everything in the world. This is it. Okay, that's all we know. When you go to school and you're in a little kid's school, you are taught this much of knowledge, this little blue part of knowledge. Then you go to high school, and then you were taught, or in middle school, and then you taught a little bit more. Then you are going to university, and then you are already expanding knowledge, or your knowledge, within what we know, but already in some aspects you're knowing more than the general people. That's university. Then you go to a master's degree, and then you really focus on one aspect of knowledge, and you're pointing towards the edge, but you're still far away. And then you go into research. And this is what you get from research. You get to touch the edge of your knowledge. you read about it, you discuss papers, you write papers on the knowledge that we already have, and this is what you do. This is where you get. You only get here if you're doing research. And then, at a moment, you focus on this little tip of where the knowledge ends. And hopefully, you will, after pushing a while, bump the knowledge. You will expand it. You will know more than anyone has ever known about that subject in the world, ever. And this is the magic of research for me. This is where you really peak. When you find something that nobody else knows, that's for me very personally rewarding. And it's a contribution that you are giving to the world and everybody else. Nobody can take that away. Yeah? So this is really the pinnacle of research. It's not a lot, it doesn't look like a lot, but if thousands of people are doing this at the same time, all over the world in all areas of knowledge, that little round thing will go more and more and more. And this is really my vision of how we contribute to the world. Why would you want to do research in the first place? So it's definitely not glamour. No, it's very boring most of the time. It's definitely not the money, because if you're treating patients, you probably earn a lot more than if you're doing research in a lab. But it's because you're probably a curious person that enjoys finding out about things. If you're not that kind of person, probably it's not for you, which is fine. And also, because you're a creative and you have a drive to think more than what you already know. You're a creative person. You want to create. So these are two, I think, very important requirements. You have to be curious, and you have to be somewhat creative. What will research really bring you then? Well, it will bring you the ability to think outside the box, that's for sure. It will give you, hopefully, personal reward, so intellectual personal reward. It will give you an excellent network of contacts. So thanks to research, I know people all over the world in my field, all over. And that's really good, because if I need some help from someone else, I can just call or send an email, and they can do the same. And it's really a global network of knowledge in my area. So it's not everything, but if you're just a doctor in your hospital and you don't know more than the people you work with, this is something else that your profession can also bring you. And it will give you additional skills that are not only the typical doctor skills. So it's not just knowing how to palpate the abdomen or whatever, or operate, but it's also math, statistics, it's epidemiology in general. Depending on your area of research, it can give you lab skills. And it's also interesting to learn different things other than your basic everyday stuff. So it's really a juggling. If you want to do research, you're juggling with your clinical practice and other things. And you for sure develop critical thinking. It's for sure your records, your registrations will be better. You're probably a more organized person and you are fulfilled. So I'm almost finished, but I want to mention a couple of things. This is something that I think is going to be all over the place very shortly. And this is machine learning and artificial intelligence algorithms. So we are now at the point where our electronic health records are so good that they can generate evidence for us almost automatically through artificial intelligence and machine learning. So you're putting in data, huge amounts of data, like big data. And this data is feeding back to you what you should be doing and what you should not be doing. Instead of doing a sample of patients in a controlled setting like a traditional trial, this is something else. This is big data in a massive scale, global scale. So this is going to come very shortly, and we need to be able to understand how much of this we can interpret, because we don't want machines to take all the decisions for us. But at the same time, we want to take full advantage of the technology that we have. So this is something that will be interesting in the future. So if you wish to succeed, so the question I was asked in the first place is how to bring these two things together. No easy answer there, sorry. But if you want to succeed in clinical research, you first want to get rid of that barrier that you cannot do something. You cannot change this. You cannot, it's like a lot of people think like this, especially in hospitals. It's impossible. Can't do it. So you first need to get rid of that idea. You can do it. You also need to be really flexible. So if you're rigid in your mind, you're probably not going to do good research. You need to be rigorous. You need to be very careful in what you do, but you also need to be flexible. And you need to be able to have a plan A, B, C for every project. Not everything works. I mean, I've done enormous amounts of work in my time that have created nothing. That's kind of frustrating when you spend maybe a couple of months on a single project that doesn't work. No, nothing comes out of that. Yeah, you need to be flexible, you need to be resilient, and you need to work. You need to work a lot. That's also the key here. I mean, you've probably been involved, I don't know, I know a couple of you, I know what you've done. I know that you probably think it's really boring at times, but it is the base. I mean, it's not all fun. It's a lot of hard work. It's a lot of sometimes repetitive, boring, not intellectually stimulating work. It's sometimes very challenging for yourself to be able to continue doing this, but you need to look at the big picture at the end and to think, if I or somebody else doesn't collect this data, if they don't do all these measurements or whatever, you're never going to get to the end. You're never going to get the answers. So you need to work hard and be resilient, knowing that in a proportion of times, you will be successful at achieving that little bump in knowledge that is the driving force of research for me. And that's basically what I had to say. I don't know if you want to discuss any of this with me, or any questions, anything you'd like to know about my work or whatever, or my path until this point. Anything you're curious about? Yeah, sure.
How did you actually get into research? I mean, after graduation, and what is the path that you have to get?
Yeah. It's a very good point, because it's not obvious, and it really depends on where you are in the world, where you work. So I've worked in the Netherlands, and that's where I actually did my first big research. And the reason why I ended up doing this is because the Dutch are really organized in research, really organized. So the whole machine is already put in place, and you just walk into a moving train, and then when you're finished, you walk out and the train is still moving. So it's very easy there. If you want to be a surgeon in the Netherlands, you're very unlikely to be selected unless you've done research. Why would a surgeon need to do research to be chosen to be a surgeon? Well, I can think of a few reasons. And even if you don't proceed with a clinical research career, it's important that you critically know how to critically read a paper. You know what importance research is, so you don't block research in your practice, because it gets in the way. But if you've been through the stages, even if you don't do it, you're not opposing it. You're facilitating others to do it. So the Netherlands have, I think, really the ideal system for research, where research people are usually full-time, at least in their early stage, so they have more time, and they're paid. That's something else that's really important. In countries like here, and probably most of the world, that's not the case. I mean, if you're a clinician and you want to do research, you do it on your own time, and you do it for free. You do it because you like it and nobody's paying you for it. How do you find your place? So what you need to do if you want to do research is you need to identify the people who will have the opportunities to give you projects. And those people exist. I mean, if you go to usually the universities have some sort of page where they have ongoing research. And usually there, some professors will tell you, I'm recruiting people. I want people to work on this project or that project. And it's a good way to start. If you do it early, I think I was a late start. So I didn't start doing research until I was almost finished with my specialty. And I regret it because I wish I had the opportunity to do it earlier. So if you do it during your medical school, of course it's added work, but it's also a time where you're more free, and you're more available maybe, and it's a good way to promote yourself, to be at curricular level, at a higher level, then you will have more career opportunities, I suppose. So it's not an easy question of how do you do it. You need to find institutions, and then at a later stage, if you want to continue doing research, you need to be working in a place that protects you a little bit, that gives you specific dedicated time, that will give you infrastructure, that will allow you to develop research and have a board administration that thinks it's important. If you don't have this, don't do research. Find another place, because you will be very disappointed, you will be very frustrated in your life. If everything is fighting you, not good. So you need to have the umbrella structure to protect you to do this. And a lot of institutions nowadays, they recognize the need. It's also prestigious for the institutions to have their name on papers and to be able to, show the whatever in the scale of institutions that they are a renowned teaching institution. So probably you want to go to one of those if you want to do research. Yeah? Anybody else? Did I convince you with that little... Yeah. Okay, well, that's it for me then. Thanks a lot. I hope you enjoyed that, and if you want to ask me any questions, you have my email there. So, yeah, good chance, and I hope you have a good future, a good career. Huh?
Thank you, Professor Frederico, for the amazing lecture. I think everyone got convinced with clinical research now on. So now we are passing to the 4 students from our 4 incomings. We will start with Leonie and Andrea from Malta, and then with Anna and Sarah from Slovenia. Thank you.
I don't know Portuguese. I don't know what's going on. Okay. So, hi. My name's Andrea Schweiger. I'm from Malta and I study at the University of Malta. I just finished my third year of medical school and now I'm going to my fourth year. So, during SCORE research, my project title is Secondary Prevention of Peripheral Artery Disease in Patients Undergoing Open Surgical or Endovascular Vascularization. My tutors are Dr. Federico and Dr. Rita, and I'm under the guidance of Dr. Elena as well. So the objective of the study is to identify prescription patterns and compliance of patients who have peripheral artery disease. And a bit of the background, so the study has been going on since 2020, and it has been noted that when patients have, for example, strokes or transcriminal attacks, they are given aggressive medication to try and prevent secondary events. However, when this comes to peripheral artery disease, this is not always the case, unfortunately. So, some techniques and methods used in the study are that the subjects have already established peripheral artery disease, and the study is retrospective. So, there's already an established database of patients that will be studied upon, and no further ones will be added. The data will be collected in a predefined database. So again, we're collecting data from these patients who are already defined. So the project outcome is actually to see if there is a difference in the patients who are prescribed different types of medications. My job in the data collection is data collection, data organization, and data inputting. So this is the type of data I collect. So they have a lot of generic ones, so ID and name, gender, age, and the risk factors that the patient may have. And then we have medication. So this consists of seeing the medications that the patient has when they are admitted to hospital. So if they're taking any statins or any antiplatelets, and the antiplatelets we consider are clopidogrel and aspirin. So if they're taking one, two, or both of them at the same time. Then we input the medications that they are given on discharge. So if they're given statins and antiplatelets, or if the medications are changed, and then 6 months later, if they actually continue with their medications, or if they stop them. So that is my job, basically. It's very interesting. It's we use S-Clinical, which is the software of the hospital. The hospital is Santa Marta Hospital, which is also a teaching hospital. And eventually what the study will show is that, well, probably there is a difference in the patients who actually continue and are compliant with the medication, and that it does prevent further events from occurring. And after the collection of this data, more data will be collected by other people regarding any other problems that the patient might have had during their hospitalization or after. So that was a very short presentation, but that's basically what the project and research is about. Thank you.
Hi, I'm Leonie, and this is my presentation about the SCORE Research Exchange. So, basically, just a little bit about me. I'm Leonie Mifsud, I'm a third year medical student, and I just finished my third year, and I'm going to start my fourth year in the next semester. Basically, I planned on going on another research exchange last year, but that had to get cancelled because of COVID. So luckily this year I was able to come here to Portugal and do my research exchange over here. And this is me at Belém enjoying a little bit of Portugal as well. So, okay, this is the title of my research project. So it's image surveillance stratification following an endovascular aneurysm repair based on the first post-operative CT angiography under the guidance of Dr. Federico and Dr. Rita. So basically, the overall aim of the project is this. So after any procedure, including the EVAR procedures, patients will obviously are at risk of developing certain amounts of complications. And obviously, to detect these complications and then be able to treat, the patients are undergoing frequent imaging. But the likeliness of developing complications wouldn't be equal in all sorts of patients that are undergoing the EVAR procedure. So the overall aim of this research is to be able to stratify patients based on whether they're likely to have, whether they're at high risk or at low risk of developing post-operative complications using a CT angiography which is taken within 30 days of their EVAR procedure. And the whole point is to be able to stratify whether they're high risk or low risk, because patients which are low risk would be getting little or even no benefit at all from continuous imaging. So a little bit about the technique and the methods used. The patient cohort is those patients which were treated for a AAA within this institution since 2011, who also had CT angiography done within 30 days of their EVAR procedure. So that's the patient cohort, which was already collected during my research. And the ones in bold are the ones which I'm kind of working on right now, which is taking the necessary measurements from the available CT angiographies and to input, obviously, this data. Once all this is done, there will be, obviously, the measurement of correlations between the findings, which I would have collected, and the outcomes of the patients. So I'm going to try to explain a bit what I'm doing. So I'm using, obviously, PACS to download the necessary imaging, and then we're using this product, Interventional, which helps us to collect the measurements necessary. So basically, you first start off with this, obviously, is the vessel with the stent inside, and you start by segmenting the vessel. After that is done, you would have to create this sort of center line where you have to check that everything is in the middle in all of the vessels to be able to kind of stretch out the vessel. And then these are the measurements which I am taking. So the proximal sealing zone, the right distal sealing zone, the left distal sealing zone, and the maximum diameter of the aneurysm. And here I have little examples. So this one is how we measure the maximal diameter. And these are basically the sealing zone. This is the proximal one, the right distal, and then the left distal. So basically you have to measure how the length of which it is sealed before it starts to detach from the actual wall. Because the longer the sealing is, the less likely that the patient will develop then complications. So they would be therefore at low risk, probably needing therefore less imaging in the future. So those were my little examples. I don't know if anyone has any questions. If not, I would just like to thank you for your time.
original. Okay. Okay. Okay, hi everyone. We're Sara and Anna. We're from Slovenia. We're sixth-year medical students, and we spent the past month researching the risk factors in patients with resected liver metastasis. So as an introduction, we've actually read about some articles about colorectal cancer, which is the third most common cancer, and the liver is the most common site of the metastasis, as you can see on pictures. 14 to 18% of patients with colorectal cancer have liver metastasis, and when they're treated, more than 50% of them have metastasis in the liver. Our study was a retrospective study, and in the end, we actually had only 45 consecutive patients with liver metastasis. We were actually researching the patients in the year 2015, and most of them were male, actually 82.2% and 70.8% were female. And the mean age was 64, which was expected. So the limitations of this study were, of course, that we only spent here 1 month, and the second one was that language barrier, because the patient's database was in Portuguese and we only speak English, so it was really time consuming to get through all the data with the patients.
Okay, so in addition to the data Anna just presented, we also checked for the most common place for the primary colorectal tumor to appear, and we found out that the most common place was the left colon in 46.7%, followed by the rectum in 15.6%. In the patient's files, we also looked for the TNM classifications. We found out that in majority of cases, the tumors were in stage T3, that was in 48.9%, and some of them were in stage T4, that was in 22.2%. Nodes were usually N1 or N2 in 28.9%. We were also interested whether the liver metastases were synchronous or metachronous, and we found out that most of them were synchronous in 71.1%. In addition to that, we also checked whether the liver metastases were unilateral or bilateral regarding the liver segments. And we found out that most of them were unilateral in 51.1%. Finally, we checked what kind of treatments the patients received, and we found out that only 7 patients received neoadjuvant chemotherapy, and 7 patients received adjuvant chemotherapy. In the end, we did the SCORE tests, and we found out that only one of all this data was statistically significant, and it was that one of the liver metastasis locations. We found out that if the liver metastases were unilateral at the beginning, they had a lesser chance of recurrence. So the ones that were bilateral did not recur in only 8%, and the ones that were unilateral did not recur in 37.8% of the cases. And that's it from our side.
Thank you.
Thank you.
Thank you, girls. Now we are doing a short break, and we will be back in a few moments to listen one more lecture and some presentations of our students. Thank you.
Yes, yes, we're here.
Hi everyone again. So now we will do another lecture, now this time with Professor Claudia Almeida, Alzheimer, what has already been discovered and what is yet to be discovered. Thank you. It's very nice to give a talk in person. It has been, I don't know, more than a year. So very nice to be here. I think I will point with the mouse so that everybody can see. Oh, wait. How do I choose the... Uh-huh. The mouse keeps on disappearing. Sim, sim, se me quiser ajudar. Eu quero carregar ali. Pois, é isso que é o meu problema. Ele está ali, mas... Ah, agora cheguei lá. Sim, vou só pôr aqui em ponteiro laser e aqui. Sim, e duplicar. Okay, now let's see if I manage to present this properly. So although I was asked to give a little, to give a talk on what is known about Alzheimer's disease and what is there yet to know, this is an impossible mission in 40 minutes. I have to say that right now, like this week, there is, it's happening International Alzheimer's meeting in Denver and in virtually, so a little bit like we are doing today, this hybrid mode, which maybe will be the future. And it makes sense actually, because it's nice. We can look in the eyes to the people we're talking to, and everybody around the world can listen. So they have more than 100 countries, people from 100 countries participating in this meeting. And they are giving an update for what is being discovered and what is yet to be discovered for a whole week. And it runs from 3 p.m. until 11 in the night, Lisbon time. So you can see how difficult it is in 40 minutes to tell you about everything that is going on. So I'll give you a little bit about, I'll talk a bit of a general introduction first about one issue that I think we still have a lot to discover, which is why do we develop dementia when we get old, and why is aging important? And this is an area of intense research. We already know a few things, but it's an important aspect. And of course, this is true for other diseases. It's not only for dementia, but it's maybe one of the most critical disorders nowadays that affect the aging population, and it's becoming even a more preoccupating problem because there's more people getting old. Because with the benefits of medicine, people are surviving a lot of illnesses, and they can actually survive for longer. So it's really becoming now a problem, the increasing numbers of people that develop dementia. So this is in some data from 2018, so it's already outdated, that 1 in 14 people over 65 have dementia, and this number rises to 1 in 6 over 80. And it's especially worse for women. That can be 1 in 3 women. So if we get to live until 85, so we are 1, 2, 3, 4, 5, 6, 7, 8. So you see 2 to 3 of us will have Alzheimer's if we get that age. So why does aging increase the risk of dementia? I mean, this is really scary, right? I mean, this is something for you to think about. So that's why I'm bringing it up. So the main form of dementia is Alzheimer's disease. Although this is a very big bag,
And this is also a problem. A lot of people that have dementia are diagnosed as having Alzheimer's disease. Because a lot of the phenotypes fit in this big bag of phenotypes fit in this biological definition of Alzheimer's disease that can only be really definitely identified post-mortem, so by looking at the brain and seeing the presence of the amyloid plaques and the tau tangles. But just yesterday I was hearing about this, that the classical or the pure Alzheimer's disease is relatively rare. Most of the time when they look at people that have been diagnosed with Alzheimer's disease, there's many other things going on in the brain. And this is one of the big problems we have right now, is that as doing science and research, we need to simplify and reduce our variables. I think the future will be increasing the variables, as when we know more about the parts, then we can try to make the puzzle and putting it all together. So some of the things that we already know is that for the most simpler let's say, cases with better understood etiology are the rare cases that have an earlier onset, so they appear before 65 years old, but this is classified as a rare disease because it's very, very uncommon. Most neurologists here in Portugal that I talk with, they have never seen a rare case of Alzheimer's disease. So just for you to have an idea, it's really not very common. But in these patients, we already understand why do they have Alzheimer's disease. And the reason is because they have mutations in either the amyloid precursor protein, so the protein that gives rise to the amyloid that forms the plaques that accumulate in the brain, or the gene is duplicated, or maybe more common, mutations in presenilins, which are the catalytic components of enzymes that cleave the amyloid precursor protein to produce beta-amyloid. So they are all linked, these mutations in the rare forms, to the amyloid production. But in the late onset, so the one which is more common that develops after 65, it's not still understood what causes Alzheimer's disease. And probably like in cancer, we'll in the future get to the conclusion that there's many different types of Alzheimer's disease with many different causes, and maybe there's not just one thing causing Alzheimer's disease. But commonly is considered that aging, genetic risk, but also environment, lifestyle, etc., are all contributing to the development of Alzheimer's disease. So as I was saying, so the familial cases are less than 1%. The mutations change the amyloid production. So the amyloid is a very small peptide with 38 to 42 amino acids. And it's cleaved out of a bigger protein, the amyloid precursor protein. And it's cleaved in the N-terminal by base, which is a sigla for beta-secretase. And some of the APP mutations, what they do is to kind of make APP more interesting for base. So it's more cleaved, and there's more, the quantity of beta-amyloid just starts being produced excessively. But with the more common mutations, the presenilin mutations, it's actually a bit different. It's not so much the quantity, but the quality. So the presenilins cleave this in the C-terminus and do it sequentially. So the shorter forms, so the 38 and 40, are less toxic and actually are the ones that are normally produced. Although the function is still not super clear, but it may have some physiological role. But the longer forms accumulate in Alzheimer's and are more toxic are these 42, 43. And the reason is more that this progressive cleavage is impaired, and so these longer forms accumulate. And just having this extra 2 or 3 amino acids at the end of the amyloid makes it more hydrophobic, meaning that it likes to stick to each other and form these aggregates, which are very toxic. They interfere with almost any function that has been studied in the cell. They are really, really nasty. And I think when people have these mutations, although you have them all your life for the longest time, you can eliminate beta-amyloid. But, of course, with aging, the capacity to eliminate these toxic forms decrease. The other thing that I wanted to mention in terms of another difficulty that has arisen relatively recently, so for the last 10 years, researchers started to realize that actually the mechanisms, the pathological mechanisms that will lead to Alzheimer's disease go by undetected for the longest time. So people had already known that once you are diagnosed with Alzheimer's disease, you progressively decline your cognitive capacity, your memory declines quickly, and with time, eventually, you become you lose all your capacity to take care of yourself, and many patients end up bedridden and incapable of anything. But what was not really understood was that the pathological mechanisms were already occurring in the brain even 30 years before the disease diagnosis. So before the person has sufficient complaints that does the tests and is labeled, you have Alzheimer's disease. So stuff is diagnosed when you're 65 or over 65, but has been going on since you were 35 or 30. So this is quite impressive. And this was only possible by doing longitudinal studies. And this is something that it's still missing. There's still a lot to be done in this area. So in this case, these were patients that were carriers of mutations that were known to, that it was known that in the future they would develop Alzheimer's disease, and they started to following them since they were 30, and already they could detect 30 years before the onset amyloid accumulation in the brain. So this is also another challenge in the field, is how can we study and treat when these problems are undetected for the most people, right? So this is another big challenge. The other thing that I wanted to make out with this slide, another message, is that there is this time course of problems which start with the amyloid accumulation and end up with the tauopathy. So the accumulation of aggregated tau occurs subsequently to the amyloid accumulation and is now known to be absolutely required for neurodegeneration to cause disease. At least experimentally in mice that model the disease that have both amyloid and both tau, experiments were done where the tau gene was knocked out, removed, and the mice got better, even though they had a lot of amyloid. And it's also known that there are people that have a lot of amyloid in their brains and still their cognition is not severely affected. It's another problem that we still cannot explain, is why is it that if beta-amyloid is so toxic, some people resist having this accumulation, at least in the form of the amyloid plaques? One answer to this question is that maybe the plaques are not the most toxic entity in Alzheimer's. It was, at least in experimental models, it was seen that it's the smaller aggregates of amyloid that are more reactive, the oligomers. And also, those go undetectable in the clinics. So the amyloid plaques are the only ones visible in the PET imaging of a patient with Alzheimer's disease. So although experimentally we know and we can follow these things happening, we still don't have ways of doing it in humans. So this is also a problem, how to bring the medicine closer to biomedical research so that we can help patients in the future. So for you, if you're studying for medicine, is something for you to worry about for our kids. Okay, so beta-amyloid precedes tau. And besides this idea that you have oligomers before you have plaques, there's another issue that I want to address, which is that it's usually defined Alzheimer's as having extracellular plaques and intracellular tangles. And another thing that goes without being analyzed in the clinics, but it's more and more considered, is that the earliest changes actually occur inside cells. So amyloid is produced inside neurons, in endosomes, and it can accumulate especially close to the synapses. And the synapses are the connection sites between neurons that allow information to flow and for the brain to function. So why am I interested in where does beta-amyloid gets produced? Because since it's the production, how it starts accumulating, I thought, okay, if we understand well how it gets produced, maybe we can interfere with things from the beginning, right? And it's very complex. So all these arrows show that for beta-amyloid to be produced on endosomes that you can see here in the middle, 3 things need to happen. The amyloid precursor protein, which is a transmembrane protein, needs to meet base, which I told you about, the beta secretase, which also is a transmembrane protein, so it's continuously moving around in the cell depending on membrane traffic. And also the gamma secretase is composed of transmembrane proteins, and the gamma secretase is the one that contains the presenilins that I told you that were cleaving on the C-terminal. So the 3 of them need to meet in the same cellular location in order for the APP to be processed. And not only they have to be in the same cellular location, but the pH of that location needs to be right in order for the enzymatic activity of these enzymes. And so the place where that is favored is the endosomes. And the endocytosis of APP is required for this production, and some also of the other ones. So APP is produced during at endosomes and during the endosomal maturation. So here you have represented what I've just told you. You have APP, the full-length APP with the C-terminus, the A-beta domain, and the N-terminus, and the sequential cleavage that ends up releasing A-beta. And this is showing an EM with an endosome with beta-amyloid accumulation in a normal brain. Okay, so this is also something else that maybe some of you don't know, is that the amyloid production occurs even in normal cells, just to a much less extent than in a disease situation. So we wanted to try to understand, so before I tell you about what we wanted to do, so besides understanding what causes amyloid accumulation, the other important aspect is if it's causing problems to synapses that are so important for establishing memory. And in experimental models of the disease, different ones, it was shown that A-beta can interfere with the function of synapses. It doesn't necessarily kill the neurons, it just causes synapse dysfunction and eventually synapse loss. But what about in the late onset Alzheimer's disease? So this is where it's not understood if increased A-beta for amyloid, or sometimes I say A-beta, which is kind of short for beta-amyloid. Does this, is it also the first event, or there's something else before? And so we are interested, actually in the lab, we work on these two aspects, the importance of aging and of genetic risk factors. Because, and I'll just mention this for the genetic risk factors, this is another big area of research right now, because the geneticists got so much better on looking at our genomes that they started to analyze the genome of patients with Alzheimer's and controls, of course, and see what was different in the genes. Were there differences that correlated with the disease status? And in fact, they found, not only for Alzheimer's, but for many different diseases, several candidates of risk factors. So there's a huge effort right now in the community to try to understand what does it mean to have these risk factors. How are they going to start the disease? What are the problems that they're going to cause? Because if we understand what they do, then we can try to correct them before we can do gene editing in humans, of course, which maybe in the future will be just a good solution. But for the process of aging, it's a bit more complex. And there's several evidence that say that already during normal aging, there's already increased beta accumulation, especially intracellularly. As in these references that I have here, it correlates this accumulation with age. And another aspect that it's important to think about is that even in the absence of disease, we all know and we are all afraid that when we get older, our cognitive capacity is going to decline. And you'll see, probably not you, but maybe your parents or your grandparents, sometimes they start forgetting things and you start saying, oh, you're getting Alzheimer's, right? Everybody kind of jokes about that. But although definitely aging is not the same thing as Alzheimer's, but maybe there are processes during aging that potentiate or facilitate the appearance of Alzheimer's disease. And maybe some manifestation of this is already the cognitive decline that people start suffering when they're older. So the good news is that there's an enormous variability about this aging cognitive decline. That means that not everybody will show cognitive decline, and not everybody will show the same extent of decline. One of the best things we can do to prevent cognitive decline is what you are doing here, paying attention to this talk and thinking about what I'm saying. Well, not only that, but having a higher levels of studying and thinking and reasoning and maintaining an active mind is been shown experimentally that it's protective for cognitive decline associated with aging. But one of the things that happens during aging is that our neurons are also aging. So most of the neurons in our brain are born when we are born. Although more recently it was discovered that neurogenesis still occurs in the adults, it's a very small fraction. It does not account for all the neurons we have. There's no turnover of neurons. We are just born with what we have. We lose some, and a lot of connections are eliminated as during infancy and adolescence. But we are stuck with the neurons we were born with. And so our aging happens to them as well. So there's no renovation. So what is the impact of the cellular aging to neuronal function is something that we are very interested in. See what I just did? I went through a very big thing. So how aging increases the chance of getting dementia. Now I'm just focusing on the cellular aging of one brain cell type, which are neurons. Not to say that aging is not important for the other cell types, but most of the other ones have some rate of replication, and neurons are very sensitive because they are so highly differentiated and complex. So we decided to ask the simplest questions that we could think of, which is why does aging lead to intracellular beta-amyloid accumulation? And if there is cognitive decline, does this A-beta accumulation have anything to do with it? And so we formulated these two questions and we decided to address them experimentally. And this led actually to another problem that we still don't have the solution, which is how do we study aging? And it's a problem because it takes a long time, and most researchers need to make their careers during their lifetime, right? So we cannot wait too long. Most of the time, even when we do mouse models, we try to do very aggressive mouse models so that they can give results quickly, and so we can publish papers and, you know, move science forward at a faster pace than nature so we can understand disease. And so we thought about how to study this, and we came up with two models of aging. Oh, I forgot to mention this. Okay, I'll just skip it. Okay. Which are, so one is a mouse that ages in 18 months. So by 18 months, they show signs of aging, including cognitive decline and beta-amyloid accumulation. The other one is because we wanted to establish causality. You know, when you have a problem, you fix the problem and you see if it solves it, right? So if we can experimentally manipulate it more quickly, we decided to use a cellular system that had been described in the literature by several groups that used neurons in culture, and you just let them age in culture. And actually, they show signs of cellular aging. Of course, it's not because of the organism aging, it's an artificial triggering of a program that each cell knows what they have to do when conditions arise that lead to cellular senescence. And so what had been described as some markers of aging are lipofuscin accumulation, which is just a product of non-degraded stuff in lysosomes, increased oxidative stress, synapse decline, so several characteristics that are also seen in vivo. And so we used these two models to ask our questions. So the first thing we did was to characterize these neurons. So can we replicate in our lab what other people had described? And so we use these primary mouse neurons. So why are they primary? Because they are obtained from the brains of mice. We dissociate the brains in their cells, and we put them in culture with a culture media that favors neuronal development, and we don't have a lot of glia or anything like that. And the cells in 3 weeks, they are maturely differentiated, they establish synapses, and they look good, they are not dying, they don't show gross degeneration. They have a senescence marker accumulating, and they have lysosomes containing lipofuscin. So I'll just skip some slides because they are just kind of details, maybe not too important for today's talk. So primary neurons, they show some signs of aging in culture. Now, do they recapitulate the intracellular beta-amyloid? That's what we wanted to study. Why do aged neurons accumulate amyloid? And in fact, they do. Not everywhere. We saw that the accumulation was, so we can see by immunofluorescence beta-amyloid with an antibody that recognizes specifically A-beta-42. And you see these deposits, these white blobs, they are more present in the older cultures than in the younger ones. We have ways of quantifying this specifically. We see that also the amyloid precursor protein is more accumulating in the neurites. And we did some controls, which were to do these types of labelings, but in cells that do not express amyloid precursor protein or amyloid. And basically, we set up our system. So we have a cell model of neuronal aging that recapitulate features of in vivo. Aging markers, a beta-42, and the amyloid precursor protein increased in the aged cultures. So why does it increase? So the first step in that big trafficking scheme that I showed you was the endocytosis of the amyloid precursor protein. So that's where we started. And to look at the APP processing, because that's the process that leads to the beta production. And making a story short, so basically we saw more processing. So we see more products of processing when the cells are aged compared to the younger ones. We see that the enzymes were not significantly altered. We saw that the endosomes where the processing occurred were increased, at least the marker of these endosomes was increased. In vivo, we saw similar phenotype, more processing, and more markers of the early endosomes. When we looked at the early endosomes, we saw, in fact, more endosomes, more APP on endosomes, especially early and not so much late. In vivo, when we looked in the brain, we could also detect more APP on endosomes. So we see here in the colocalization, there's more colocalization between APP and EA1 at 18 months of age. So there's more processing, early endosomes are increased, so is there more endocytosis? So endocytosis is the process of moving APP into the endosomes. And we use a technique with the biotinylated proteins. Can explain if you want in the questions. And we saw a lot more endocytosis, so darker bands means more biotin being endocytosed, more APP getting in. And by immunofluorescence, we also see the same. These are endosomes containing APP, and you see a lot more when the cells are just older. We don't do any treatment to the cells, just looking at them, and they are just endocytosing more APP and making more beta-amyloid. So after we went to try to understand why this is happening, and we did a bunch of controls looking at different things, but just to give you kind of the nutshell, we saw that not only APP endocytosis was increased, so if we look at a general marker of endocytosis called bulk endocytosis, we see also increased endosomes, but the differences were bigger for APP. But another marker of endocytosis was decreased, and then we were like, okay, this is really strange. So the general marker is increased, APP is increased, but this one is decreased. Oh, maybe we discovered something new. Maybe this is a specific decrease of this marker. So what is the marker? The marker is transferrin that binds iron and transports it into cells. And iron changes have been associated with aging. So it could be another phenotype of the cells that they don't need iron, so they just don't endocytose it. So we look at other markers that were, again, supporting our first evidence that there was an increase in endocytosis. Then we blocked the mechanism of endocytosis with different drugs. One of them, it's interesting, chloropromazine, is actually used to treat patients for schizophrenia. So we started to think, okay, so can we reverse APP endocytosis with this? This could be a treatment possibility. And we did. We blocked endocytosis of APP. And so we need to explore now for the future if this would also reduce beta-amyloid production and eventually what is the impact on synapses. So I'll just skip this because it's just more details for you, I think. I can get a little bit into the mechanism if you want. But basically we showed that neurons, just by getting older, they show an upregulation of endocytosis of APP that could underlie or at least contribute to the accumulation of beta-amyloid in the brain during aging. So is this increased production synaptotoxic was our second question. And of course, I wouldn't be saying it, asking the question if we didn't find the answer. So we did see that the total levels of synaptic proteins were not unchanged in these conditions, which is different, for example, from an Alzheimer cellular model where we would see, I had done this work in the past, and we saw significant reductions of glutamate receptors, for example. And we saw that the aged cultures had less synapses. So how did we do that? We colocalized the pre and the post-synaptic. So the yellow dots represent synapses here at 21 days in culture, the younger neurons. And you see less of these yellow dots here, so it means the synapse number reduced. And then we treated these neurons with a drug that reduced beta-amyloid, and we saw more synapses. And we did this with 2 different drugs, and basically we concluded that beta-amyloid accumulation in aged neurons contributes to synapse loss. Probably is not the only reason why there's synaptic decline, but for sure it's not innocuous. So it is doing something bad to neurons. And the other experiment that we did after was the opposite. So we showed that aged neurons did more endocytosis, more beta-amyloid, and less synapses. Now, if we do it experimentally, if we increase endocytosis on purpose, like not by aging, but just us, we just do it. We do a treatment to the neurons that makes them endocytose more. So can we age them faster? That's what we asked. And that was the case. So when we overexpressed RAP5, which is a RAP GTPase, that it's a major regulator of endocytosis, so you have more endocytosis going on. We saw more beta-amyloid in the neurons, and we saw less synapses. So in fact, we could make neurons age faster. What we have not been able to do is to make them age slower, which would be what we would like, right? But that's something that we want to investigate in the future, and that's our next step. There was also a paper that came out more or less at the same time as ours that did a mouse where they used the same strategy of overexpressing RAP5. So they make a mouse that has this overexpression. What do they see? They saw also degeneration, less spines, less LTDs, cognitive deficits, even tau changes, endosomes accumulation. So in a way, supporting that downstream of the changes that we saw very early in neurons, when translating into the mouse, and with time, they build up and they cause significant problems. So I think we nailed down a very important mechanism of aging. So these are my conclusions, but I think I already went really long. So I'll just conclude saying that we published and we made the cover of Journal of Cell Science. So if you want to read, it came out in May this year. And finally, I would like to say that Tatiana was really the drive force of this project. She worked on this project, just for you to have an idea, for 6 years until we managed to get a story ready for publication. And this is only possible with funding by many agencies that contribute to our lab and to our research. So I just would like to thank you for being here in this live talk and hybrid visual one. I don't know who's on the other side, but I am happy to take your questions. Ta-da! Yes, there's already a question. Yeah.
I actually have 2 questions.
Mm-hmm.
First is, since the amyloid is getting
I didn't understand, sorry.
Are there actually some comorbidities?
No, this part I understood, but the part about the beta-amyloid in the beginning,
Maybe about amyloid depositing in some other parts, not only the neurons.
oh, I see. So the neurons are the cells that express more beta secretase, and so they are the main producers of beta-amyloid. I've seen some publications that say that beta-amyloid can be produced by other cells, but when we look in the brain, it's in neurons that we see the intracellular accumulation of beta-amyloid. But beta-amyloid, when it's produced in neurons, it accumulates in endosomes, but it is also secreted to the extracellular space. And when you have aggregates in the extracellular space, there are other brain cells like glia, like the astrocytes and the microglia, that help clear out the amyloid from the brain by endocytosing this beta-amyloid. But they are very good on digesting it, and they eliminate it, okay? So we do not see aggregates forming inside these other cells. The amyloid plaques are extracellular, and we still don't know well where they come from. So the idea is that some people defend that there are neurons that are accumulating so much, they form dystrophic neurites, and at a certain point, these neurites kind of burst out and release these intracellular aggregates, and they seed the plaques that then, more stuff comes into and starts aggregating and form these very big plaques. This happens in the brain, right? And because the brain is relatively contained from the rest of the body, there's not a lot done by accumulation in other tissues. But there is passage of beta-amyloid through the blood-brain barrier. There are transporters like the lipoprotein receptors, for example, that bind, and beta-amyloid can bind to lipoproteins too. So they can bind and help transpose beta-amyloid into the circulation, and then it can be eliminated in the liver. Maybe, I have not thought about this, so this is a good point. So I don't know if the people that die with Alzheimer's, if they would look in other tissues, would there be, for example, in the liver accumulation? One thing that we know is that when there's a lot of aggregation of beta-amyloid in the brain, you actually see less of this passage to the circulation. So one of the markers, maybe that's why we don't see, one of the markers of disease progression is actually a decrease of beta-amyloid in the CSF and also in the blood. So it's more retained in the brain. So I would say not so much.
Okay, and the second question was, are there some particular areas of the brain where the amyloid tends to deposit, or is it just casual?
Well, I think some people have looked at this very carefully, and they've seen that the hippocampus and some regions in the hippocampus and the temporal lobe are more, or are the earliest sites where you see amyloid deposition. And then with time, there is accumulation in the synaptic connected areas first. So it's almost like it follows the circuit with time in the brain. But it's not very restrict, like for example with Parkinson's disease, it's more restrict to the dopaminergic substantia nigra. But in Alzheimer's, it's not as restricted, and it's very spread out through the whole cortex very quickly. Okay? Welcome. Thank you. No question. The online people don't make any questions? No? Okay. So on my first page, there was also my email, and I'm also on Twitter, so if you want to ask me questions later, you can. Thank you so much. I'm just going to take out my talk. Right.
Thank you, Professor Claudia. It was an amazing lecture. Alzheimer is a big world, and 40 minutes it's a little bit.
A big challenge, right?
Yes. So thank you for this amazing lecture.
Good luck with everything.
Thank you.
Bye-bye.
So now we are passing to our last three students, and now we will have Elena from Russia and Biljana from Bulgaria. They are via Zoom with us, and they will present their research. And after them, we will have Francesca that is here with us. So thank you.
Do you hear me? Is the connection okay? So, hello dear colleagues. Today me and Elena are going to present the project that we participated in pancreatic duodenectomy decision tree machine learning for patients with pancreatic ductal adenocarcinoma of the head of the pancreas. In this project, we worked alongside Dr. Emanuel Vigia and Dr. Catarina Aguar. I'm sorry if it's not the right pronunciation. We worked at the Curry Cabral Hospital here in Lisbon. So first, I would like to do a quick recap of the anatomy of the pancreas. I know that you all know it, but just a quick review. So, as you all know, the pancreas is a blunt-shaped organ positioned at the level of the transpyloric plane. It's a mainly retroperitoneal organ with the exclusion of the tail of the pancreas. It consists of a head, neck, body, tail, and a foot-neck processes. It's an exocrine organ, and mainly it consists of system of ducts. The main pancreatic ductus runs along the length of the pancreas, and it combines with the bile duct, which drains in the duodenum via the major duodenal papilla. The main disease that we face in the pancreas is the pancreatic cancers, and the main neoplasms that we face is the ductal adenocarcinoma in almost 80% of the cases. There are also cystic neoplasms, tumor carcinoma, islet cells neoplasms, and metastasis, but the main one is ductal adenocarcinoma. You can see in this picture the different types, and the last one is the adenocarcinoma. You can see the destruction it does to the cells of the organ. The main risk factors that connected with this disease are smoking, diabetes, chronic pancreatitis, obesity, also old age and family history. And unfortunately, also gender is more common with men than with women. So one of the main problems that we face is that the diagnosis of pancreatic cancer comes too late because in the early stages, it's an asymptomatic disease. And when the symptoms arise, it's mostly late for the patient. Common symptoms are fatigue, jaundice, loss of appetite, weight loss, different color of the stools and the urine. And of course, that if a patient with these symptoms comes, we do diagnostic tests like evaluation of the alkaline phosphatase and bilirubin, because they can be increased if there is a bile duct obstruction or liver metastasis. Normally, the amylase and lipase levels are normal. We do imaging with CT, MRI with contrast, also ultrasonography, and of course, we look for increasing the tumor markers. The main one is CA19-9. The treatment, here is the main opposition between surgical and non-surgical treatment in the early stages, because there might be a better outcome for the patients if there is first treatment with radiation and chemotherapy and not surgical one. But in the later stages, it's mainly surgical, and the Whipple procedure is the surgery that is done. It's a pancreatic duodenectomy. It's a removal of the gallbladder, the duodenum, the head of the pancreas, and then the small intestine is attached to the stomach, the bile ductus, and the remaining part of the pancreas, as you can see here in the picture. Of course, there is also symptomatic control, analgesics, usually opioids if there's pain, also pancreatic enzyme supplementation. The prognosis of this disease, unfortunately, is poor. In the 5 years of survival, it's less than 2%, because most patients, as I said, are diagnosed later on, and the tumors also can grow back. The good thing is that up to 10% of the patients who receive early diagnosis become disease-free after treatment. So that is one of the main goals, to be able to diagnose patients early in the onset of disease. So now Elena will tell you more about what we did in our project.
Thank you so much. What we wanted to do in this research, we wanted to create a simple and practical nomogram for decision if a patient with pancreatic ductal adenocarcinoma of pancreatic head benefits of a pancreatic duodenectomy, which shows satisfactory predictive efficacy. Next, please. Okay, the prognosis of patients with pancreatic ductal adenocarcinoma, or PDAC, is pancreatic head remains poor even after potentially curative RO resection, because the 5-year survival rate for PDAC is only about 8% for resectable PDAC of pancreatic head. And pancreatic duodenectomy remains the major treatment option. However, patients following curative resection have different outcome due to tumor heterogeneity. Next, please. The aim of this study was to develop an accurate model to predict patients' prognosis for pancreatic ductal adenocarcinoma of pancreatic head following pancreatic duodenectomy and its real benefit. We asked a question: which patients really benefit of a pancreatic duodenectomy? Next, please. Our team has been following up 397 patients with PDAC of pancreatic head from January 2004 to December 2019. Those patients underwent pancreatic duodenectomy with RO or RA resection at Curie Cabral Hospital. All the patients that had received the optimal chemotherapy by synthesizing the patient's tumor burden, physical and financial condition. What we excluded was a refusal to follow up, patients diagnosed with preoperative infection, hematological or inflammatory diseases, patients with history of other malignant cancers, no informed consent, and patients with unknown origins or distant metastasis. The protocol of this study was submitted at the Clinical Research Ethics Committee, and patients' clinical pathological characteristics were collected from medical records with a lot of parameters, such as gender, age, different kind of chemotherapy, different kind of invasion. We also were checking blood tests within preoperative days. We paid a close attention to neutrophils, platelets, lymphocytes, ALT, AST, ALP, albumin, total bilirubin, and tumor markers CA99 and CA125. All the patients with pancreatic ductal adenocarcinoma in our hospital were followed up by our team. Patients routinely underwent enhanced abdominal CT scans or MRI in every 3 months within the first year and every 6 months thereafter. The objective survival is defined as the period from the period between surgery and confirmed death or final follow-up. We will be using several statistical methods that you can see on the slide. Feature selection is conducted to identify the subset of parameters that most contributed to the design of the predictive models, and all experiments will be conducted using a machine learning open source software, the Orange Data Mining. What do we expect? We expect to create a decision tree machine learning based on patients with pancreatic ductal adenocarcinoma of the pancreatic head that helps to define which patient will benefit of a pancreatic duodenectomy. We also expect to develop a novel prognostic model to predict the objective survival and the relapse-free survival for patients with pancreatic ductal adenocarcinoma of the pancreatic head following curative operation. And we also expect to establish a nomogram incorporating features collected that presents high predictive accuracy, and all in all, to improve overall surviving and qualities of patients with PDAC. In conclusion, we wanted to say that the most important aim is to avoid unnecessary treatments with high morbidity and mortality to maintain patients with quality of life. Thank you so much. Thanks.
I'm Francesca, I'm from Italy, and these past few weeks I've been working in the Department of Interventional Radiology, and my tutor was Professor Tiago Belim, and I was helped by Dr. Ines Condevasco, and the title of the project is Single Center Retrospective Analysis Comparing Bidimensional Angiography Machine with Combined CT and Angiography Machine. I was working in the Hospital of Curi Cabral in Lisbon. So first of all, I'd like to introduce what interventional radiology is. Basically, interventional radiology is a medical subspecialty where the doctors can use the radiological images such as X-ray or magnetic resonance and ultrasound to perform minimally invasive surgery. And they perform little incisions. Usually the patient is awake, and they use catheter tubes, needles, wires to perform both diagnostic and therapeutic procedures. Diagnostic such as biopsies, ultrasound images, or therapeutic procedures, for example, for the treatment of tumors, of gastrointestinal diseases, genitourinary diseases, embolizations, and this kind of procedures. For the first week, I've been in the OR with the doctors and could see some of the procedures that they perform. One important procedure is the chemoembolization. The chemoembolization is a treatment for the hepatocellular carcinoma, so for the carcinoma of the liver, but there are some criteria to choose and select the patients that can undergo the treatment. As I already said, it's a minimally invasive procedure, so the doctor gets into the femoral or radial artery with a little incision, and then uses the catheters, as I already said, some needles, and use the guide of the real-time X-ray, which they can see on the screen, and can reach, following the vessels, can reach the vessels of the liver and then the vessels that are feeding the tumor, so that they can perform something selective or super selective, and they can inject, once they are in the vessels of the tumor, they can inject a drug which is made of microspheres loaded with doxorubicin, which is a chemotherapeutic. And like this, they can both make a vascular occlusion and a chemotherapy, a selective chemotherapy, which lets the patient avoid the bad side effects of the systemic chemotherapy. And it's also important to remember that there is a maximum dosage they can inject in the vessels, so they might have to perform many sessions of this treatment, usually once a month. So you can see, for example, the image on the right, which is the X-ray image, and you can see the little catheter and then the vessels that are feeding the lesion. This is where the doctors inject the drug so that they can select the vessels and then treat only the lesion. Another important procedure they perform is the ablation. The ablation consists of a needle which is put through the skin and reaches the lesion, which is seen with the X-ray image or the ultrasound image, as you can see on the right. And it can be both a radiofrequency ablation or a microwave ablation. A radiofrequency ablation is made with currents that pass through an electrode and then create the region of heat to destroy the cancer cells, while the microwave ablation is made of microwaves that are created by the needle, and as before, they create a region of heat to destroy the liver cancer cells. This is the room where they perform their procedures. As you can see, there is the X-ray machine on the top, which can be moved, and it's important to remember that it is used in real time, so the doctor can see the images on the screen on the right and can see exactly where the catheter is, how he should move to reach the right vessels, and he can also inject the contrast to see perfectly the vessels. And then in the back you can see the CT machine, which was put in the room in 2018, and it's really important because it's the aim of my project. So going to the investigation, what was the investigation about? The investigation, as I already said, has to do with the CT machine that was put there in 2018. We are collecting and trying to analyze the data of the patients which were treated between October 2018 and September 2018. We are analyzing all the patients' data, but in particular we are trying to focus on the chemoembolizations that I explained before and the ablations. And what we are collecting is, of course, the personal data of the patient, the radiation exposure, which is really important for interventional radiology, since the radiation is used a lot to perform the surgeries, and the time of the procedures, and also, of course, the outcome of the procedures. So what we are trying to understand is we have the bidimensional X-ray angiography on the left and the three-dimensional CT with the X-ray overlapped together on the right. And we are trying to understand, is it better if we use a three-dimensional CT overlapped on the X-ray in real time or not? Does it make a better outcome or not? So on the left, you can see, as I already said, the angiography with the X-ray. On the right, you can see the CT three-dimensional image, which is overlapped to the X-ray in real time. You can see perfectly the vessels, you can see the catheter here, and then you can see this little blue ball, which is the representation of the lesion. So the doctors just, as I already explained, put the catheter in the vessels, reach the liver vessels. Once they reach the liver vessels, they perform a CT scan, and like this they can see where the catheter is and where the lesion is and the vessels that are feeding the tumor. Once they do that, they overlap the images, and this is what they see on the screen. On the left, the X-ray image that is moving while the doctor moves the catheter. On the right, the overlapped images. So you can see the catheter, which is going to move while the doctor is performing the surgery, and the CT scan they performed before. Like this, they understand where the vessels are, which are the vessels that they have to select. And of course, on the underside, you can see, of course, the parameters of the patient. And here you can see a little bit of contrast that is being put through the vessels. So the big question we are trying to answer to is, does the CT angiography machine provide a better treatment efficacy? Does it provide less radiation exposure, less time of the operation? Is it easier for the doctor to reach the right vessels or not? We are collecting the data, and then we will, in the future, analyze them, compare them, and try to understand and quantify this issue. Thank you for your attention.
Unfortunately, due to some unforeseen with the speaker of the third lecture, we will have to end our symposium without this lecture. I would like to finish by thanking everyone who allowed this symposium to be held. To incomings, by the impressive work, and to their tutors, to the amazing speakers, Professor Frederico and Professora Claudia, to the university for the space, and to all the staff of communication department from the university for the brilliant work with the streaming logistic, to the management nucleus of ISM for all their support, with special thanks to Afonso Andrade, to the communication department of ISM for the excellent work with the events image, and to my international department colleagues, Margarida and Diana, as also to the Nori, Rui Pancas. Thank you. I hope you liked the symposium.